Hello, my name is Jordan Berg. I'm from the University of Utah, and I'll be presenting my talk titled Gazing into the Metaboverse, Automated Exploration and Contextualization of Metabolic Data. First off, I would like to thank the organizers for the opportunity to speak to you all today about our new tool for metabolism data analysis, Metaboverse. But before we get too into detail with this tool, I want you to imagine you have never heard the story of Little Red Riding Hood. How difficult would it be to grasp the story with these sparse details? Is that a wolf or a bear? Would you know Little Red Riding Hood's grandmother was eaten by a wolf? I fear this is often how we tell the story of metabolism. Metabolism is a complex, integrated network of reactions that fuel the biological processes needed to sustain life. And contextualizing the data can often be very challenging, even for experts in the field. As an example, let's look at these steady-state metabolomics data of paired normal and adenocarcinomic lung tissue samples. Standard approaches might begin with plotting the data like so in order to prioritize hits. But this could miss important relationships regarding how these components work together to form this complex cooperative network, particularly if these patterns emerge in pathways we are not as familiar with. We therefore developed Metaboverse to allow users to integrate multi-omic datasets onto over 90 model organisms. While network approaches for metabolism may not be new, we introduce features that automate complex pattern recognition across the network and prioritize regulatory patterns to lead to a more robust regulatory experience. For each omics type, whether it be sequencing, proteomics, or metabolomics, log fold changes and statistical values appropriate for the data type are provided. In this case, we will provide metabolomics values for this data set. But now we are faced with a challenge. Yes, we could manually search through the network and canonical pathways for interesting patterns that would be missed by the volcano plot shown previously. But how do we create a more holistic experience for the user? To automate this process, we developed an automated regulatory pattern search engine. This engine searches the global reaction network as annotated by the Reactome knowledge base for patterns such as the one on the right, and returns these motifs prioritized by the user-provided statistics. Here we see a reaction where the measured input is increased in concentration, while the measured output is decreasing in concentration. This pattern may indicate some regulatory event occurring across this reaction that would be of interest to the user. This pattern search engine can be further informed by other reaction components, such as catalysts and inhibitors. Additionally, since we take this global approach, this feature can operate across reactions between pathways that may normally be missed by canonical representations of metabolism. Let's narrow in on the example just shown. While the authors of the original study were able to identify broad upregulation of nucleotide metabolism and other features, they were unable to identify this particular pattern. This reaction, where glyceric acid is normally converted to 3-phosphoglyceric acid, has close connections to serine metabolism, a pathway frequently dysregulated in cancer. This pattern may therefore indicate some regulatory mechanism here that feeds into dysregulated serine metabolism and may contribute to tumorigenesis. Moreover, this event is easily discernible using Metaboverse. Additionally, if a user finds a metabolite or protein of interest and would like to explore the upstream causes or downstream consequences of these changes across the reaction network, they can search one or several of that component's reaction neighborhoods. Users can also identify cross-compartment motifs based on the hole shading demonstrated in this image. However, often a given metabolomics dataset may only measure 100 to 200 metabolites, while there are over 1,000 involved in metabolism. In order to address this challenge, we developed a method of summarizing reactions with missing data by merging these reactions if the terminal ends of the subpathway are measured. For an example, if reaction A has measured inputs and outputs, this reaction is maintained as is in the network. But in reaction B, where it's missing measurements for an intermediate step, these two reactions can be merged to generate one pseudo-reaction, thus allowing for more complex pattern searching across this space. While we are continuing to develop this algorithm, we see a simple example of the utility here in the lung dataset when looking at the conversion of L-serine to pyruvate. This transition can occur by two methods, one a one-step process and the other a two-step process. This reaction collapse allows for automated discovery of regulatory patterns across both types of conversions. It could then be interesting to follow this up with RNA-seq or proteomics to determine which subpathway is being utilized and whether this was biologically relevant. Another feature of metabolic networks, and networks in general, is connectivity. The consequences of network connectivity in biology are straightforward. You remove, for example, a transport reaction of a vital metabolite, which can be signified by the dashed line, and the downstream reaction chain breaks down. Within Metaboverse, we can explore perturbation network from the dataset by stitching together all the reactions with at least one perturbed component. We can see that while there are components of metabolism where several subsequent reactions are perturbed, others are more independent perturbations. This may lead us to hypothesize that drugs targeting more connected reactions may not be quite as viable as targeting more disconnected reactions where there is less redundancy in the chain. In summary, Metaboverse provides a user-friendly interface for better contextualizing metabolic data by automating complex pattern detection within the metabolic network, along with allowing users to explore the consequences and extent of these perturbations on the network. Using biological data, we were able to identify known and unknown regulatory events that then allow us to generate new and interesting hypotheses for further follow-up and validation. I would like to thank the entire Metaboverse team and the Rudder Lab for their contributions to this project, along with our funding sources. But in particular, I would like to thank my mentors, Jared Rudder and Bei Wang, as well as Cameron Waller, who played a substantial role in the conceptualization of this project, and Yu Jiazhou, who played a critical role in the design and implementation of the pattern search engine. To learn more about Metaboverse, you can access our preprint and the slide deck from this presentation through the provided QR code. Thank you for your attention, and I look forward to any questions.